Alright, biology, this is Mr. B, and we are getting ready to start our final uh, section here in Chapter 1, and this section is entitled Tools and Procedures. So far we've kind of discussed what scientists do, kind of what we look at as biologists in particular, so now we're going to kind of look at some tools that are going to help us out when we're studying biology. So, first thing that's kind of a big idea in this chapter is a measurement system. So what measurement system do most scientists use? So when we think in our daily lives, we use things like inches and feet and miles and things like that. However, that kind of could be a problem because scientists all around the world need a common system of measurements in order to replicate each other's experiments. Again, we have to be able to always test and retest experiments, and if we're using one set of measurements over here, then scientists in France isn't going to be able to perform the same experiment. So scientists across the world have decided to use the metric system when collecting data and performing experiments. So whenever we talk about experiments running in class, we're never going to use inches. We're always going to talk about um, the types of measurements that lie within the metric system. And actually, every other country in the world pretty much uses this metric system besides us. So the metric system is a decimal system of measurement whose units are based on certain physical standards and are scaled on multiples of 10. So because a metric system is based on multiples of 10, it's easy to use. So we'll go ahead and look at the chart here really quick. So you've probably heard some of these terms before. The most common one would be a length. So 1 meter equals 100 centimeters, and then 1 meter also equals 1,000 millimeters, and 1,000 meters equals 1 kilometer. So when we think of how we use inches and feet, like 12 inches equals 1 foot, like 5,280 feet equal 1 mile, all those numbers can get really confusing. It's a lot easier to just remember that all of this falls into a multiple of 10. So the metric system is a lot more popular because we have a lot easier multiples to use. So just remember, if you ever want to be a famous scientist one day, you're going to have to learn this metric system and learn how to use it. So moving on away from, I guess, our measurements, we're going to look at some tools that we have. So the first tool, and it's a really big tool, are microscopes. And these are devices that produce magnified images of structures that are too small to see with the unaided eye. So we have two types of microscopes. We have light microscopes that produce magnified images by focusing visible light rays. And if we ever use microscopes in class, these would be the ones we would use. And then there are electron microscopes, and these produce magnified images by focusing beams of electrons. So electron microscopes are only going to be found in the big science labs. They used to look at very extremely tiny things. So let's take a little more in-depth look at light microscopes. These are the most commonly used microscope, and they produce clear images of objects about a magnification of about 1,000 times. So that's decent. This is what, again, kind of standard what we have. So as well as a light microscope, there's kind of been an improvement called the compound light microscope, and these allow light to pass through the specimen using two lenses to form an image. And this allows us to magnify it a little bit bigger than what we would with just a regular light microscope, having these two lenses. So light microscopes make it possible to study dead organisms and their parts and to observe some small organisms and cells while they are still alive. So this is the most, I guess, effective one that is actually practical to have in a laboratory setting. However, again, like I said, there's limitations. Light microscopes cannot produce clear images of objects smaller than 0.2 micrometers, or about 1/15th the diameter of a typical cell. So if we want to look at those smaller things, get an in-depth look at maybe some viruses, which are going to be a lot smaller than a normal cell, we need to have a different type of microscope. So that's why scientists use electron microscopes. So again, electron microscopes use beams of electron rather than light to produce images. The best electron microscopes can produce images almost 1,000 times more detailed than light microscopes can. So this is, again, the big dog, I guess, in terms of microscopes. It's the one that's really going to be the most effective. However, it's also going to be the most expensive, so you're not really going to see them as often. So electron microscopes. So getting away from material, we're going to look at some techniques that scientists use. Scientists use two techniques maybe to look at cells, which is going to be our next unit. The first one is called a cell culture, and a cell culture is developing a kind of a big group of cells so we can study them more in depth. It's kind of hard to study just a single cell sometimes, and if we need to look at a big group, we want to grow them and we want to make a bunch of them so we can study them more easily. So Scientists do this by placing a single cell into a dish containing a nutrient solution. The cell is able to reproduce so that a group of cells, called the cell culture, develops from the single original cell. So then another technique they go over is called cell fractionation, and this is just used by scientists, by biologists in specific, to break down cells so they can just look at different parts. So if I only want to look at my nucleus of a cell, we can use cell fractionation to break down the cell and isolate only the nucleus. So those are two techniques that they mention. Maybe be able to know the difference between these two if you ever saw them on the test. So really quickly, by wrapping up this tools and procedures, when we're working in a lab, we always need to consider safety. So scientists working in laboratory or in the field are trained to use safe procedures. And laboratory work may involve contact with dangerous materials or organisms. I hit on this a little bit on the first day about how we need to be safe, and again, I'll give you more training when the time comes on how to be safe. So kind of some general practices to follow would be study the safety rules. Again, just look over so you know exactly what to do. Read all steps and safety precautions. Follow your teacher's instructions and textbook directions exactly. And then if in doubt, ask your teacher for an explanation. Again, that's a big one. Don't ever just assume you know how to do anything, or if you're scared and don't know what you're doing, again, don't be afraid to ask. And then wash your hands thoroughly with soap and water after every scientific activity. Again, we talked about how there's trillions of microorganisms out there and probably in the room as we're working. We don't want to get any of that inside of our body. And then the big thing is when working safely in biology is remember that you are responsible for your own safety as well as that of your teacher, classmates, and any live animals you handle. So this is again going back to being responsible, being mature in the laboratory, because there's not just yourself in the risk, it's a lot of other people around you. So that's it for this chapter. Let me know if you have any questions.